Thank you very much, Professor Hollander, for the introduction. And first of all, I'd like to say that I feel very honored to be here today and talk after people who have actually shaped their fields with major breakthroughs during their career. And even though it is very challenging to speak after Professor Wood, I think it's the perfect link, because a lot of things that he said in his presentation, you will see that actually make total sense with the continuity of my presentation. So what I'm gonna talk about is how we can accelerate the drug development for neurogenetic conditions. We're gonna talk about neurogenetic conditions, which most of them are actually rare diseases. So I graduated in medicine in 2016, and shortly after that I arrived in the UK, where I worked as a junior doctor in general pediatrics and neonatal units in big tertiary centers of the country. And during that time, I came to some realizations, and I would like to share these realizations with you in numbers, which can be overwhelming, but it is important to mention them because these are the numbers that actually fuel with passion our work. And what I'd like to say is basically that 30% of the children who are admitted in pediatric wards are actually affected by a genetic condition. And Professor Wood gave you the statistics about the US, but I'll give you the statistics about the UK right now. And 3.5 million in the UK are actually affected by rare disease. And 80% of the rare diseases are actually genetic diseases. Now, most of the people who are affected with a rare disease will undergo a diagnostic odyssey from the first manifestations of their disease until the actual diagnosis. And this will involve a lot of specialist appointments and a lot of investigations. Now, we are in front of a new era, and the first reason is the fact that we can now diagnose a lot of conditions that have remained largely undiagnosed in the past. And that's thanks to newborn screening programs and thanks to genome sequencing programs. And neurological conditions or neurodevelopmental conditions have remained for many years some of the conditions which we did not have a known genetic cause, but after the whole genome sequencing, now we are in a position to say that we can have we have identified the cause in the genetic code of these patients. And these are the results of a pilot study that has been done under the umbrella of the 1,000 Genomes Project, 100,000 Genome Project, where, as you can see, the neurological or neurodevelopmental conditions, especially if we combine those with those who have intellectual disability, had some of the highest diagnostic yields. So now we are in a position to identify causes for diseases or conditions that remained undiagnosed, but also we have to consider that we have to act in a timely manner. And here I will present you very quickly, there are of course some periods during brain development, which basically for some model organisms have been identified during the first, as for example, for Drosophila melanogaster, we know that there is a critical period during early development, and this accounts for the approximately first 5 years of life for kids. And we know that that there are some mechanisms involved that make these periods having heightened plasticity, but this, unfortunately, is not the same later on in life. And this shows that we need to act early, and regardless, and of course, there is a lot of research that is happening at the moment of on the mechanism. This was a big part of my work before coming here in Oxford, of trying to identify the molecular mechanisms of reopening critical periods, but we don't know if we will actually be in a position to do that. Therefore, we need to act early. And why we need to act early now? And that's the most important question, because now we are in front of a new era of treatments for genetic conditions. And we're in a new era because we have changed the way we approach the treatment of these diseases. So when someone has a genetic condition, they usually have an alteration on their genetic code, the DNA, which we call a mutation. And this leads to lack or abnormal function of a protein. And this has some results, the combination of which we call a disease. So basically, so far, the approach was that we were trying to tackle some of the results or a combination of these results. But right now, our approach has changed, and we are mostly aiming to tackle these diseases at DNA or RNA level. And that is particularly important because it is actually why we are talking about an interventional treatment. We are talking about tackling diseases at DNA or RNA level and actually treating the root cause of these diseases. And this, as Professor Wood also mentioned before, and Professor Servet has actually played a very important role for, is the Spiranza, the nusinersen, which has been approved in 2016 for spinal muscular atrophy. And here I will show you a video that Professor Servet has very nicely offered to me about two patients who have been treated at different time points. And these two patients are actually siblings. So this means that they actually have exactly the same disease when it comes to genetic background. And the girl that you can see there has been treated before the initiation of symptoms, whereas the boy has been treated after symptoms. And that was because they were aware of the fact that the boy had the disease because he was older, so they have tested on time for the girl and they have given the treatment timely. And that is particularly important because it shows the importance of having newborn screening methods in place. And for that reason, the STRONG group, led by Professor Servet, has already established a program, and now in Thames Valley, newborns are screened for spinal muscular atrophy, and we hope that in the next few years, or maybe it is happening right now, this disease will be eliminated in the whole area. And of course, there are other diseases for which major breakthroughs have happened in the last 2-3 decades. A lot of therapies that are aiming to target, potential therapies that are aiming to target DNA or RNA level are at preclinical or clinical development. And some of these breakthroughs have happened here in Oxford within the department in Professor Wood's lab. And we have seen that we have not reached exactly the exact same dream as we have reached for spinal muscular atrophy, but we have a lot of tools towards this. And of course, we have to be careful because these decades of development have actually shown us what are the important, have shown us some lessons. What are the important steps that we need to take, the action that we need to put in place in order to avoid mistakes that have happened before in the drug development for these diseases. And basically, the thing is that the drug development for these diseases can be sometimes impaired. It can have obstacles that are other than the efficacy of the drug. This is not very common, but we need to take into consideration why sometimes we need to act on time. So basically, in 2020, I arrived in Oxford, where I started my PhD in Paediatrics. And I'm supervised by Professor Servet and by Professor Rinaldi. And I have to say that I feel very lucky to be supervised by these two very inspiring men. And they complete very nicely my training from bench to bedside. And I joined the STRONG group with the dream, with a mission to accelerate the drug development process for rare diseases. So the STRONG group is led by Professor Servet. And I was told that before actually entering the group and start any work, I needed to show some proof that I can run the 10K for the Muscular Dystrophy UK. And that's a precondition, always. And after that, I was allowed, of course, in the group to start some work. And despite the fact that the group has a huge experience in accelerating drug development for neuromuscular diseases, we decided to expand our limits and go to other diseases which are not neuromuscular, but the expertise of accelerating drug development can be transferable. And one of these diseases, which is our now one, has saved a lot our work, is Angelman syndrome. So Angelman syndrome is a neurogenetic condition, so it is associated with changes in a specific area of the genetic code of the patients, one gene, the UBE3A. And children have global developmental delay, they have movement impairment, they have balance issues, they have impaired expressive communication. In many cases, so in most cases, they don't have words, but in very few cases, depending on their genetic background, they might have a few words. They are affected by seizures, and in many cases, they have sleep problems. Now, These children are really lovely, and I'm sure that they bring a lot of happiness to us, but I'm sure that they bring a lot of happiness to their families as well. That's what I've heard from all the families that I've met during this journey. But of course, they can also bring a lot of anxiety as they don't become independent adults, and they need someone to care for them around the clock, even during the night. Now, the reason why we decided to work on Angelman syndrome is because we saw that there is a huge potential of having a drug approved very soon. And that is because there are more than 15 different therapeutic approaches, and most of them, despite what has been traditionally done in the past, aim at DNA or RNA level, as I explained before. And of course, we saw the potential of avoiding mistakes of the past and accelerating drug development for this disease. And drug development, generally the whole process from the early discovery of the targets and the compounds to the clinical development and the approval looks like that, which basically looks like a very linear process, but in fact is not easy at all, and it has a huge attrition rate from the early development to the approval. And in some cases, we are talking about a 90% attrition rate from the compounds that start the development to the compounds that finish. And talking specifically about Angelman syndrome, I'm going to show you some obstacles, some hurdles that each stage can have. And these are just examples because obviously there are many more which would need a new presentation for them. So when it comes to preclinical development, it's very difficult to actually have representative models of the disease, Angelman syndrome in the specific case. And that is because a lot of different genetic backgrounds, a lot of different alterations in the genetic code of the people can actually have the same result as a disease, but they might have differences in how we need to approach a therapy for these people, or differences in what toxicity the same therapeutic approach might have for them. And that's for the preclinical development, one main obstacle. And then when it comes to the clinical development, one main obstacle is the rarity of the disease. So when it comes to clinical development, we need, especially at late phase clinical trials, a lot of participants to make sure that the results that we have we can draw statistically significant or non-significant conclusions, but we can have a safe number to draw these conclusions on. So the rarity of the disease is makes particularly challenging the clinical development for diseases which have a lot of drugs at the same time at development. And this is because inclusion in one of the clinical trials can actually lead to patients to be excluded from other clinical trials. And this can be overcomplicated by the variability of the phenotype that I explained to you before, the fact that different genetic backgrounds can actually create the same disease phenotypically. And of course, what we need to do to make sure that we accelerate drug development, clinical development, to make sure that we have the right tools to assess the efficacy of these drugs, but more importantly, not just the efficacy of the drugs, but what makes sense for the families, for the people who actually receive these drugs. And today we see that we have a huge lack of outcome measures and biomarkers that can be objective, sensitive, and sensitive to change. And this is one of the most important fields for development when it comes to accelerate drug development for rare diseases. So we designed and set up with a strong group the natural history study for Angelman syndrome, which is funded by the Foundation for Angelman Syndrome Therapeutics UK, which is a charity run by families of patients, and this is particularly important. I will explain you why. So a natural history study is basically a study that, through a lot of complicated protocols and procedures, actually follows up patients without giving any intervention, without giving any treatment over time in specific time intervals. So the main procedures that happen in a natural history study are basically questionnaires, assessments, but also collection of biological material and a lot of other clinical investigations, as for example EEG, or other measures of clinical outputs, as for example measurements of clinical outputs, as for example using accelerometry and other digital devices to measure clinical outcomes. So what happens is that this specific study has a huge scientific value because it contributes in the development of outcome measures and biomarkers for the diseases. But as you can see here, this is one child that wears one, I can't open it, here we are. This is one child that wears, you might not be able to see it very well on this video, but on her ankle she wears two watch-like devices which actually can measure her walk, they can measure her movement in a daily setting at home in an uncontrolled environment. And this is particularly important because this type of assessment, this type of measurement can actually help us develop outcome measures which can then become eventually endpoints in clinical trials, which actually make sense for the patients and for the families. And actually one of these outcome measures is now submitted for FDA approval. And continuing from that, it has, as I said before, a huge value for the community. And we are very proud to say that this study is funded by the Foundation for Angelman Syndrome Therapeutics, and that shows that it has a huge value for the community. First of all, because us as investigators come in touch with the community, with the families, and we can learn, we can hear the voice, we can take their feedback in our work, and we can also make sure that we are prepared to create the infrastructure to accommodate clinical trials in the setting. And this, what has happened within less than a year from the start of the natural history study, we have started clinical trials here in Oxford in the UK, and we can proudly say that we have actually managed to deliver a therapy, an antisense oligonucleotide, for the first Angelman patient in the UK here in Oxford within this year. And this has a particular importance for us, but also for the community. Also, other things that can derive out of a natural history study and can help tackle hurdles that are also earlier in the development is that out of the collection of the biological material, we can isolate cells and create models of the disease in a dish where we can screen new potential treatments or actually identify biomarkers. And also, using multi-omic approaches, we can identify panel of proteins or panel of other biological molecules that can help us understand at which stage is the disease, where is the disease going, and when do we need to act with a treatment, or if the treatment is working. And of course, as I said before, maybe the drug development process looks much more linear than it really is, but in fact, it's much more complicated and requires a constant feedback between the clinical research and the basic research. And the natural history study actually can contribute in a lot of areas that are both at preclinical and at clinical setting, and can help accelerate the drug development, can accelerate the process of bringing a therapy from the bench to the bedside. And the Department of Paediatrics is particularly an amazing place to be for this specific process. It is basically the place to be for this process, and this is because it has shown that the collaboration between stakeholders at bench and at the bedside of the field of research is actually possible within the department. And this is, for example, the strong links that we have between our group and Professor Wood's group. It has also shown that almost all stages of drug development can actually take place in the department, and there is a plan of expanding, and there is a successful record of collaboration with communities and industry when this needs arises. And of course, this is particularly important to have the means to facilitate collaborations with communities and industries, because all stakeholders need to work together towards drug development. And of course, this progress is accelerated by very responsible and committed staff within the department. And with that, I would like to show you this picture, because it's actually teams that work in the Department of Paediatrics and at the Children's Hospital at the John Radcliffe. And here we can see that we can actually have the whole process of drug development in Oxford, and we have shown that we have a great record of collaborating with industry and communities. And basically, a great dream from the future would be to see the story that, as it developed for Angelman syndrome, to develop for other rare diseases, and maybe be here at the 70th birthday of the department and say that we have actually managed to take a drug out of Professor Wood's lab to the clinic via the Strong Group clinical trials. And of course, I would like to thank the Strong Group, Professor Servet for supervising me for my PhD, and Professor Carlo Rinaldi for supervising for my PhD. And I would like to thank you for your attention.
Thank you very much for this very inspiring talk. Are there any questions? Charlie. And then Chaz.
Thank you very much, Dora. I was wondering, what do you think an academic institution needs to be able to deliver drugs in rare disease, especially?
I think that basically, if I go back to my slides, I think that we can find the answer here. So basically, an academic institution, I think it needs to encompass different teams, that work together in the whole drug development process, from the very early discovery to the preclinical stages and then to clinical development. It needs to have in place the right procedures to facilitate the collaboration between communities and industry, because it's always important to make sure that we have a good collaboration with all stakeholders that are involved in clinical development. And of course, to be flexible to accommodate change, because these lessons that we learned from the drug development for rare diseases are fast. We need to have a fast pace in order to be able to accommodate the changes that we need to do and the adjustments in terms of facilities, in terms of changing practice, in terms of being ready generally.
Thank you. That was superb. I mean, the messages I've got from yourself and Matthew is that there's no doubt in the next few years you will be doing more early genetic screening. I'm sure you'll develop platforms to develop gene therapies more cheaply, more quickly, and I'm sure you'll get funds to push those into the clinic. How big a gap is the biomarkers? How do we scale that up? And, you know, some of the videos you've shown, I was almost thinking you don't need proteomic biomarker signatures. You know, it's pretty damn obvious when something works. And these are monogenetic and they're less complex than common diseases.
I agree. I think that generally it's very difficult to say that we are close, but I think that we have identified the problem from the previous years of clinical trials, and we have identified the problem that we cannot continue assessing patients with questionnaires or assessments at a single time point in time when they come for a study or at a clinic appointment. We need to have objective outcome measures and biomarkers, and outcome measures and biomarkers that can measure change in actual life, in everyday life. And of course, the sensors that you saw on the child of this girl are not there to burden her, but are there to measure what is actually meaningful for her and what is actually meaningful for her parents, because a clinical scale might not be able to capture that climbing some stairs sometimes can be life-transforming for some people. So I think we are maybe far for some diseases, for some others maybe closer, because the nature of them actually makes it easier. For example, where there is a movement impairment, then using digital technology, accelerometer, for example, can lead very quickly to some meaningful results. As, for example, we have already submitted a proposal to the FDA for Angelman syndrome, but in other cases we might need to work on more on other ways, like identifying biological molecules or panels of biological molecules that can show us the way.
Thank you very much.